Okay, hello everyone. Good morning, Jay Shri. Good morning. Okay, what about others? Good morning, Sahani. Good morning. Okay, just a moment. Good morning, Sumaya. Good morning. Okay, let's try to understand this concept of sensory photobiology. Okay, good morning, Savita. Okay, in this particular sensory photobiology, what kind of questions we can expect, friends? Good morning, Lakshmi. In the sensory photobiology, friends, we can get the experimental based questions. Experimental based questions, then friends, followed by the statement related questions. These are all the only two ways where we can expect the questions from this particular sensory photobiology. Mostly, friends, in the part B I am talking about. In the part B we can get the statement related questions, friends, wherein it comes to the part C we can get the experimental based questions. So we have to know this concept in terms of experiments, also in terms of statement based also. Good morning everyone, please continue. Okay, so listen, actually in this chapter we have to read about three pigments very majorly. Good morning everyone. So we have to read about the phytochromes, we have to read about the cryptochromes, and we have to read about the phototropins. So these are all the three concepts, friends, we have to read very elaboratively in this particular sensory photobiology. Good morning everyone, please continue. Okay, now let's start. Good morning. Okay. So listen, once again I'm telling you before starting, that concept is very easy. I'll make it more simple, but I want you to remember maximum amount of the information to answer the questions very easily. Okay, now let's start. Let me explain you 15 to 20 minutes continuously. Automatically I'll solve your doubts. Photobiology, friends, listen, how can I define the word photobiology? Photo, light, biology, the branch of biological science. Everyone listen, photobiology is the branch of the biological science which involves the interaction of the light with living organisms. Everyone listen, how living organisms are going to interact with the light for the purpose of growth and development. That is what exactly we can call photobiology. Now you can ask me, sir, why exactly plants need the light? What are the various uses? So all of you please listen, friends. It can be used as the source of energy for the process of photosynthesis. Not only that, friends, it can also act as the source of information. See, anyway, source of energy for photosynthesis, we all people know that where the plants are going to convert the light energy into chemical energy, and it starts using for the process of photosynthesis to prepare the food. That's, we all people know that. Not only for photosynthesis, friends, plants also starts using the light for the process of phototropism, for the process of photoperiodism, and for the process of photomorphogenesis. So in the entire chapter of the sensory photobiology, we are going to read phototropism, photoperiodism, and photomorphogenesis. Friends, listen the lesson very carefully. By tomorrow morning this entire chapter will get over. I will solve all the previous year questions, so you have to answer very easily. So make sure that you listen the lesson properly. Now listen, now in this entire lesson we are going to read these three processes: tropism, periodism, and photomorphogenesis. Let's try to understand one by one elaboratively, friends. Listen to me carefully. Photomorphogenesis. So what exactly the word photomorphogenesis? how can I define the word photomorphogenesis? Listen to me carefully, friends. Light signals regulate the changes in structure and form of the plants. So all the students listen. Why exactly light is required for the plant body? All of you listen to me. For the process of seed germination, for the process of leaf expansion, and friends, for the process of flower initiation. For all these purposes, what do we require, friends? We require the light. So all these listen to me very carefully. All these changes, all these processes occurred with the help of a light. That is what exactly, friends, we can call it as a photomorphogenesis. So the process of seed germination, the process of leaf expansion, the process of flower initiation, which is exclusively mediated by the light, that is what exactly, friends, we can call the photomorphogenesis. Okay, who will do all this? Undoubtedly, friends, all this can be promoted by a particular type of a pigments. What do we call it, that particular pigments? We call it that particular pigments as the photoreceptors. So friends, the question comes here, how did we categorize the plant pigments actually? Every one of you listen to me carefully, friends. The plant pigments are broadly categorized into two types. One we called mass pigment. Friends, listen to me for 15-20 minutes, automatically you will understand. Once we read one or two slides, we don't get a clear cut idea. The plant pigments are broadly categorized into mass pigments and sensor pigment. Okay, based on what we categorize the plant pigments into two types. One is, friends, how much amount of the light it is absorbing, first point. Second point, depending on their concentration, whether they are present in the lower concentration, whether they are present in the higher concentration. Based on this, we have explained it actually. Mass pigments, sensor pigments. Mass pigments, name itself indicating they are present in the high concentration, there is no doubt about it. At the same time, they are going to absorb a very larger fraction of the incident light. And what is the best example of the mass pigments which are present high concentration in the plant body, friends? Undoubtedly chlorophylls, as well as, friends, carotenoid. Kindly do not forget it. Now friends, when you look into the sensor pigments, what you have to read about the sensor pigments, it absorb only a small fraction of incident light. Very small fraction of incident light it will absorb. And friends, plant pigments are not at all present in high concentration. When compared to the mass pigment, friends, their concentration will be extremely lower, very lower, right? And what are the best example? Photoreceptors. Now we people, we never read about the chlorophylls and carotenoids because this is not the chapter we have to read. We already know, we already read the chlorophylls and carotenoids in the photosynthesis. What we have to read in this chapter of sensory photobiology is completely about the photoreceptors. This is what we have to read. Now everyone understand, all the photoreceptors present in the plant body categorized into different types. Once again, based on what parameter, what friends, based on what parameter we categorize it into many types, friends, based on how much amount of the light they are absorbing, at what wavelength they are absorbing the light, at what fraction they are absorbing the light. Based on the light absorbed, friends, we categorized all the photoreceptors into phytochromes, then followed by, friends, blue photoreceptors. I want you to remember all the three pigments. The entire lesson of the sensory photobiology is exclusively studying about these three pigments, structure, functions. Most important, the experiments. We have to read only these three. Phytochromes, red, they basically absorb the red and far red light. That's why, friends, phytochrome exhibited in the PR form. PR represents phytochrome, absorb red light. PFR form, phytochrome, absorb far red light. Blue photoreceptors, the best examples are cryptochrome and photochrome. So I'm so sorry, this is not a photochrome actually, it's a phototropin. It's not a photochrome friends, it's a phototropin. Okay, just listen to me carefully. We have to read structure of these three pigments. We have to read the structure of the cryptochrome, phytochrome, phototropin. We have to read the functions also. Most importantly friends, the genes that are basically coding for these three pigments we have to read. Okay, let's start studying the first type of the photoreceptor, that is the phytochrome. Kindly do not forget that. What is phytochrome? All the students listen. I already told you friends, it absorb red light, it absorb far red light also, but not strongly, but a little fraction, not strongly, a very little fraction. It can also absorb the blue light. Friends, the unique statement every one of you has to remember, in a given part B question or a 2-marker question, they can ask the question like this: all of the following functions are performed by the phytochrome except or which of the following function is not mediated by the phytochrome. Friends, even this kind of questions also we can expect here. So what is the ultimate function of the phytochrome? In the presence of the red light, phytochrome, listen to me carefully, exclusively responsible for flowering, exclusively responsible for seed germination. Now you will get a doubt, sir, we read in the plant hormones, hormones are the one basically responsible for the flowering, hormones are the one which is exclusively responsible for the seed germination. There is no doubt about it. Hormones required for the flowering and seed germination, but friends, along with the hormone, even light is also extremely important. Kindly don't forget that. So it mediates red light stimulated response, flowering as well as seed germination. I will explain you how phytochrome helpful in the flowering, how phytochrome helpful in the seed germination. This unit is completely about information, lot of information to be remembered. The phytochrome basically exists in the two forms. What are the two forms of the phytochromes, friends? Everyone listen. Phytochrome exhibited in the PR form, phytochrome exhibited in the PFR form also. What is PR form? P represents phytochrome, red light absorbing form. P represents phytochrome, FR far red light absorbing form. So friends, I told you that phytochrome exhibited in the PR and PFR form, right? So how? Based on what we categorized into PR form and PFR form? Of course, based on the light absorption, there is no doubt about it. But also friends, PR and PFR form, this discovery was actually based on the seed germination process. Now listen to me, every one of you, very carefully friends. Discovery of PR and PFR forms. Everyone listen. Red light, all the students look into the diagram carefully. Here it was written that red light induces the seed germination, far red light inhibit the seed germination. All the students look into the diagram. Actually these are all lettuce seeds. What seeds, friends? Lettuce seeds. You only try to understand clearly when we keep this particular lettuce seeds in the dark condition. You only try to observe, friends. Do you observe any kind of a seed germination? No, there will be no seed germination at all. But friends, whatever the lettuce seeds that we kept in the dark condition, when we exposed to the red light, once again listen to me, when we exposure to the red light, what happens, friends? It is allowing the process of seed germination. After some time, what I have done, once again I have started treating immediately with the far red light. When I exposed to the red light, seed germination is induced. When I exposed to the far red light, seed germination got inhibited. Once again, this far red light, when it is exposed to the red light, once again friends, the seed germination got stimulated. Once again, when I exposed to the far red light, seed germination got inhibited. So friends, what is the common statement that every one of you can understand here? Listen to me carefully. Whatever the function that is performed by the red light, that will be basically cancelled by the far red light. Vice versa, whatever the function performed by the far red light, the function of the far red light is actually cancelled by the red light. Here what we can understand with the help of an example, red light inducing the seed germination, it allows the seed to get germinated. So far red light is acting opposite to the red light, so it never allows the seed to get germinated. Even the seeds are already germinated, it will inhibit the process. That's why both are opposite to each other. So the unique statement you people has to remember, both the PR form and the PFR form, these both are interconvertible to each other. What friends? Both are interconvertible to each other. That is what exactly friends we can call the photoreversibility. Interconversion of the PR and PFR forms after the light absorption. Kindly do not forget it. You only kindly look into the diagram. This is the phytochrome which is in the PR form. This PR form of the phytochrome, upon absorbing the red light at 670 nanometer, it is converting into what form friends? It is converting into the PFR form. Very simple point it is. Now same way, all the students look into this. This PFR form, upon absorbing the light, it is upon absorbing which light? Upon absorbing the far red light, it is going to converted into PR form. So that's why what I can define, both the PR form and PFR form, both are interconvertible to each other. This interconvertible phenomena we call it as friends photoreversibility. Please kindly do not forget this point, the point of photoreversibility. Okay, look into this. This is not required. Okay friends, kindly don't get confused with this graph. Just listen to me carefully. Photostationary state, you know, everyone listen my words carefully. Photostationary state is a kind of a state where the spectra of the PFR and the spectra of the PR and PFR overlap. Once again, listen my statements, don't get confused at all. It's very easy. See here, this is the PR form. This is what form friends? PFR form. At one particular point, listen to me carefully friends, at one particular form, the PR and PFR get overlapped with each other. That point we can call it as a equilibrium point. What point we can call the equilibrium point? The point where the PR light and the PFR start overlapping with each other. So see, all the students listen. This is what light? Red light. This is what light? Far red light. Now everyone try to understand friends, in between the red and far red light, what is their absorption ratio? Everyone try to understand. In red light, PFR form is 85%, while PR form is 15%. Don't confuse with this. Whereas in far red light, PR form is 97%. In PFR form, very less, that is the 3%. So friends, what you have to understand, in the red light, the dominant form is the PFR. In the far red light, the dominant form is the PR. How they are dominant, we will understand in the upcoming slide. Just listen to me carefully. What is photostationary state means? It is a equilibrium state between the PR light and PFR, where the spectra or graph of the two PR and PFR overlap with each other. That is what exactly we can call the photostationary state. Every student listen my words carefully. Phytochrome structure. Friends, our part B questions will come from here. The structures. Structures are extremely important. We have to remember the structures. Okay, and not the structure, I mean to say what we can say, the structural features. Phytochrome is made up of two components. Everyone has to listen. In the tomorrow class, I will ask this only. Phytochrome contain chromophore, phytochrome contain apoprotein. So let me define you one by one. Every phytochrome contain two components. One we call chromophore, one we call apoprotein. What is chromophore? Every student listen to me carefully. Here the example of the chromophore is phytochromobilin. What was that, my friends? The example of the chromophore, phytochromobilin. Every student listen to me carefully. This is the chromophore, correct na? How many are there? 1, 2, 3, 4. What ring it is? Pyrrole ring. Friends, you people no need to remember the structure. Kindly do not worry about it. So what do you call it this as, friends? Linear tetrapyrrole. Kindly do not forget the statement what I am saying. What do you call it this as, friends? You call it this as the linear tetrapyrrole. Linear tetrapyrrole structure only we can call phytochromobilin. Now this is chromophore, but chromophore also associated with another structure, right, friends? What do you call it this particular structure? We call this particular structure as the apoprotein. What is the point I want you people to remember while we are studying this particular apoprotein? Everyone listen to me carefully, friends. This is apoprotein. Apoprotein means, friends, what is that? Polypeptide only, right? This polypeptide chain, how it is connecting to the linear tetrapyrrole? It is connecting to the linear tetrapyrrole with the help of a specialized linkase. What do you call it as? Thioether linkase. So once again, quickly understand, every phytochrome made up of two parts. One we called chromophore and another one we called, friends, apoprotein. Once again, listen to me very, very carefully. The chromophore is the linear tetrapyrrole and it contains an apoprotein. Both are connected with each other by the thioether linkages. That is what written here. And this phytochrome is in which form? PR form. Now you can tell me very easily, how does this PR form is going to be converted into PR form? PFR, I think you are unable to see the below structure, but the structure is same. Don't confuse. After you can see the structure in the PPT. PR, upon absorbing the red light, it is just converting into PFR form. Very simple, friends. PR form is the cis form. PFR form is the trans form. There is no difference at all. What you people have to remember for the exam point of view is what they are actually made up of. Chromophore, apoprotein. Chromophore is the linear tetrapyrrole. It is not a closed tetrapyrrole. If you quickly recall, friends, chlorophyll. Chlorophyll having what kind of a structure? Closed tetrapyrrole. But here, linear tetrapyrrole. Very, very simple. So PR chromophore undergoes cis-trans isomerization at the position 15th carbon and 16th carbon, and it is converted into PFR form. Very, very simple point. Majorly remember these 2 points. Now, friends, how does this phytochrome is getting synthesized? You people should not confuse. Don't think that chromophore and apoprotein joined together immediately and they can synthesize the phytochrome. That is wrong statement. Listen my words carefully. Chromophore synthesized separately. Apoprotein is also synthesized separately, but chromophore and apoprotein joined together and then only they are going to give rise to the phytochrome. So how does phytochrome is getting synthesized? And first of all, what I want you people to remember, they can ask a simple question that which of the following organelles, listen to me carefully, which of the following organelles involved in the biosynthesis of phytochrome? Every student listen, friends, there are 2 major organelles are involved, friends. One is the plastids and another one is what, friends? Cytosol. Plastids and cytosol, these 2 are the regions where the phytochrome is getting synthesized. But one more question for the part B. Which of the following can act as a precursor? Which of the following can act as a starting material for the biosynthesis of the phytochrome? The starting material we can call the 5-amino-levulinic acid. What was that? 5-amino-levulinic acid. Phytochromobilin synthesized from 5-amino-levulinic acid. Where does it occur, friends? Of course, it occurs in the plastids. Okay, what is phytochromobilin? I told you na, it is the one which is acting like a chromophore. So friends, chromophore got synthesized, no doubt about it. Now, whatever the chromophore that got synthesized, that chromophore moving from where to where? That chromophore, that is phytochromobilin, from the plastids, it entered into the cytosol. What is the reason? What is the reason it entered into the cytosol? Because in the cytosol only apoprotein is present. That's the reason. Assembly of chromophore and the apoprotein by an autocatalytic process. Friends, what do you mean by this autocatalytic process? I told you na, by the thioether linkase. So friends, what is the point every one of you has to remember? Chromophore and apoprotein not synthesized in the same location. Chromophore synthesized in the plastids. From the plastid, chromophore come to the apoprotein, combined with the apoprotein, involved in the biosynthesis of phytochrome. Kindly do not forget this. Okay, and once again the question which is asked in the exam: in which kind of a plant seedling, what type of phytochromes will be present in the abundant amounts? Every one of you listen, friends. Type 1 phytochrome, it exhibited in the PR form, PFR form. Type 2 also exhibited in the PR form, PFR form. Kindly don't worry, you no need to remember at what nanometers they are absorbing the light. You never get in the exam, even in the experimental based questions also. Type 1, it absorbed the maximum amount of light at the PR form at 666, PFR form 730, PR in the type 2 654 and PFR in the 724. You no need to remember, kindly don't worry about it. What you have to remember for the exam, which we can expect in the statements, already asked statement in the part C questions. See, in dark grown pea seedlings, friends, sometimes instead of giving the dark grown, they will use a word called as etiolated pea seedlings. In dark grown pea seedlings or the etiolated pea seedlings, type 1 is about 9 times more than the type 2. Don't forget the statement. In light grown seedlings, type 1 and type 2 are present in the equal amounts. I want every student has to remember these 2 points without fail. Dark grown or etiolated. Type 1 cannot, see, this type 1 cannot survive longer time without the light. So that's why their number, this type 1, it is more than the type 2. I'm so sorry. Type 2 cannot survive without light for the longer time, whereas type 1 can survive. I said opposite, so sorry. Type 1 can survive in the absence of light also for longer time, but not the type 2. That's why in the dark grown pea seedling, type 1 is more in number, whereas type 2 is less in number. In light grown, of course, type 1 and type 2 are present in the equal amounts. Then the last and final point, this is also asked one of the statement in the given 4-marker question. Phytochromes, especially in the Arabidopsis thaliana, jasmine-like seedlings, it is not jasmine. In Arabidopsis, phytochromes are encoded by a multi-gene family having 5 structurally related genes. See friends, it's easy to remember also. There are total, every student listen, there are total 5 phytochrome containing genes are there, only 5. A, B, C, D, E. Everyone listen, only phytochrome A can code for the type 1. When it comes to the B, C, D, E, easy to remember. These 4 are coding for the type 2 phytochrome. So friends, that is about the first type of a pigment or a photoreceptor, phytochrome. So whenever you are reading the phytochrome, what you have to understand? First thing, why the phytochrome is required for the plant body, which means their function. Then how many types of phytochrome is exhibiting, at what wavelength it is exhibiting as the PR form, at what wavelength it is exhibiting as the PFR form. Then most importantly, the structure of the phytochrome and the functions of the phytochrome, biosynthesis of the phytochrome, at final the genes coded by the phytochrome. Only these concepts I want you to remember. Is it clear, friends, everyone? Is it clear, everyone, till this slide? Please respond. If you have any kind of a doubts, you ask me, I will re-explain. Just say yes or no, everyone. I hope it's clear for everyone. Okay, now we read about the phytochrome. Now we are going to read about the two others. What are we can called cryptochrome, then followed by the phototropins. These two we have to read now. Okay, listen to me very carefully, friends. Where we can get the experimental based questions, friends. 99.99% experimental questions will come from the phototropins only, especially from the photoperiodism. This is the area where we can get the experimental based questions. Kindly don't forget it. Okay, now cryptochromes, second type of a big photoreceptor, cryptochrome. See, friends, it's very easy. Once you compare one photoreceptor with the another photoreceptor, when you keep on comparing, you can remember the more amount of the information. So friends, quickly recall what we studied about the phytochrome, what we understand, why does the phytochrome is required for the plant body. I told you very clearly, phytochrome is required for the flowering. Of course, true point. We also studied that phytochrome is also required for the process of seed germination also. This is what we people studied. It required for flowering, it required for the seed germination also. This is what I told you. Now every student, listen my words. Why does cryptochrome is required in the plant body? So every one of you listen. It is required for inhibition of the hypocortical elongation. Three major function. So you know now, friends, suppose example, this is the coleoptile. What do you call it? This part we can call it hypocortical. What do you call it? This hypocortical. And what do you call this upper part? You can call it epicortical. Correct na? Hypocortical, epicortical. So friends, this hypocortical never be elongated in the presence of cryptochrome. That's a simple statement. So cryptochrome inhibit the hypocortical elongation. Second point, the cryptochrome will involved in the cotyledon expansion. Then the third one, it is involved in the regulation of circadian rhythm. What are circadian rhythm? Biological clocks. Friends, we don't read about the biological clocks in this chapter of sensory photobiology. Where we will read about the biological clocks, we will read in the unit 11, evolution. There we will read. But I want every one of you remember for the exam point of view without fail. Once again I am stressing, most of the part B questions will be appeared from the structure as well as the function only. So inhibition of hypocortical elongation, cotyledon expansion, regulation of circadian rhythms. Don't forget it. Okay. Now let's try to understand, friends, what is the structure of the cryptochrome. How you can remember the structure of the cryptochrome very easily? By comparing with what? By comparing with the structure of the phytochrome. Friends, once you recall the structure of the phytochrome, what you use it to remember? Phytochrome contain chromophore, phytochrome contain apoprotein. Of course, there is no doubt about it. Now every student listen. Cryptochrome have two components, same like phytochrome. Cryptochrome also having two components, one we called chromophore, another one we called, friends, apoprotein. Kindly do not forget this particular statement. Now everyone of you listen. Chromophore, which is the dual chromophore. Friends, once again keep on comparing. In the phytochrome, what is the name of the chromophore we people studied? The name of the chromophore we studied is the phytochromobulin. Correct, na? This is what we have studied as the chromophore. But friends, when you look into this particular cryptochrome, it is having dual chromophores. I want you to remember this compulsory. One is FAD, another one is pterin. See friends, pterin is an organic compound. Every student listen to me, don't get confused. Pterin is an organic compound which is always associated with the molybdenum. We read about the pterin in the nitrogen metabolism chapter also, while studying an enzyme called as nitrate reductase. There we studied about the pterin. I hope you recalled it. Anyway, that's just listen. Remember, dual chromophore, one FAD and one pterin. And friends, every student has to remember for the exam in terms of part B. What is the apoprotein here? So the name of the apoprotein in the cryptochrome is called as bacterial photolyase enzyme. Friends, where we studied about this particular bacterial photolyase, we studied about the bacterial photolyase in the molecular biology chapter while reading a repair called as a photoreactivation repair or the direct repair. I have stressed this point. Bacterial photolyase is the enzyme which is first discovered DNA repair mechanism enzyme. What friends? Photolyase is the first discovered DNA repair mechanism enzyme. I want you people to remember, you please kindly don't think that the photolyase which is present in the bacteria and photolyase present in the plant, both are not same. Both are not similar. I mean say both are not same. That photolyase enzyme which is present in the bacteria involved in the DNA repair activity. But whatever the photolyase present in the plant, it is just acting like a apoprotein, friends. It did not have any kind of a DNA repair activity. It never show any kind of a activity. So please kindly do not get confused while reading. Always try to compare. It will be very easy. Now let's once again compare. How many type of phytochromes we read? Phytochrome 1 we read, phytochrome 2 we read. Same here also we are reading two cryptochromes. Cryptochrome 1, cryptochrome 2. That's a simple point. Every one of you listen. Where does this particular cryptochrome 1 is present? See, I hope you remember na what I told you, friends. Phytochrome 1 and phytochrome 2. When we are reading the phytochrome 1, I told you it can be easily survive in the dark grown conditions also, but phytochrome 2 cannot survive in the dark grown. But what we read here, cryptochrome 1, it is stable in the light, which means high intensity of light. What friends? High intensity of light. Even though how much intensity light is present, at higher concentration, at high intensity also, Cry1 can withstand easily, but Cry2 is not like that. It require actually very low light intensity. You know friends, even the light intensity is medium also. Forgot about very higher. Even the light intensity is medium, or if it is more, it will be completely degraded in the light. Now friends, every student listen to me carefully without confusion. Cry1, Cry2, you know, Cry1 and Cry2, they will perform functions individually also. Second point, they can also perform the functions by combining also. They can perform the function individually, different functions individually. But when they combine it, friends, they will perform different kinds of, same functions. Let me explain you. Just listen my words. Cry1 basically will perform two major functions. What is that? Friends, this is only important for the exam, don't forget. Cry1 inhibit hypocortical elongation. First point. Cry1 also involved in the regulating the circadian rhythms, which is biological clocks, which we will read in the evolution chapter. Friends, when it comes to the Cry2, what the Cry2 is going to do? It mediates blue light stimulated cotyledon expansion. Kindly don't confuse Cry2 mediating blue light stimulated cotyledon expansion. So friends, Cry2 is exclusively alone responsible for cotyledon expansion. Cry1 alone responsible for inhibition of the hypocortical elongation and regulating circadian rhythm. Whereas Cry1 and Cry2 together, this is the unique statement you all people has to remember. Cry1 and Cry2 also play a role in the flower induction. Friends, why I will always stress the points is, most of the students will read for a very long time, very long time, hours together, but they never remember. Once again, I don't mind to say this word n number of times. It is not at all important how much you read for the competitive exam. How many hours you read for the competitive exam doesn't matter. It's only matter how much you remember. More amount of information you remember, more easily you can answer the questions. So even though you read less time, try to remember maximum amount of the information. That is the best way of memorizing the concept multiple times. Always remember that. So Cry1, Cry2 together involved in the flower induction. This is what, friends, second pigment. So we read about the phytochrome, we read about the cryptochrome. Then what is the last one, friends? The last one is the phototropin. This is the most important one. From the phototropin only, friends, we can get the experimental based questions. That is the process called as photoperiodism. Let me explain you briefly here. Once again, keep on comparing with the cryptochrome and phytochrome. How many types of cryptochrome 1 is there? How many types of cryptochromes are there? Cryptochrome 1, cryptochrome 2. How many types of the phytochromes are there? Phytochrome 1, phytochrome 2. Same way. How many types of the phototropins are there? Same. Two type of phototropins are there. Phototropin 1, phototropin 2. Phototropin 1, it mediates the phototropic responses at a very low light fluence rate. Whereas phototropin 2, it basically mediates the phototropic responses where, friends, at a high light fluence rate. Kindly do not forget this point. Same way, cryptochrome 1 also can survive in the no light condition. Same way, phototropin, even though light is low, that's sufficient. It can show its response. Phototropin 2, if it want to show any kind of response, the light should be at a very high intensity. Now, friends, once again, remember my statement. While we studied the phytochrome and cryptochrome, what is the two common statement we all remember? Both phytochrome also, cryptochrome also contain same. What? It contain chromophore and it contain apoprotein. There is no change. But there is a little change in the structure of the phototropin. Every student look into the picture carefully, friends. This is this is the structure of phototropins. Phototropin contain N-terminal domain. Phototropin contain C-terminal domain. You all people can see this. It contain N-terminal domain and it contains C-terminal domain. All the students listen to me. Near the N-terminal domain, you all people can see here, there are two chromophores are there. You can see now these two are chromophores. We can call it them as a love domain, LOV1, love 1 domain, LOV2, love 2 domains. You know, inside the love domain, what is present, friends? Of course, FMN is present. What is the function of FMN? FMN only friends basically act like a chromophore. This is towards the N-terminal domain. Friends, if you look into the C-terminal domain, what I want you people to remember here, at the C-terminal domain, see, look into this, the red color one. It is having serine and threonine kinase activity. Now the point is here, this love domain can be connected to the output domain or serine threonine kinase domain with the help of a chain we can call joining alpha chain. Shortly we can call J alpha chain. So once again, please listen to me carefully. I'll re-explain. If you look into the structure of a phototropin, it's a protein, consists of N-terminal domain, C-terminal domain. This particular phototropin broadly divided into two regions. One we called photosensory region, another one we called, friends, output domain. Kindly do not forget this point. Photosensory region contain two domains, correct na? Love 1, love 2. Each love 1 and love 2 domain contains what? FMN. What is the function of FMN? Acting as a chromophore. And the red color one, you can see output domain, it is having serine and threonine kinase activity, which is connected with a chain called as J chain or J alpha chain. Kindly don't confuse with this. Now, what are the functions of the phototropins? We know what are the functions of cryptochrome. We know what are the functions of phytochrome. Now we have to understand the functions of phototropins. There are multiple functions are there. First let me explain you. Phototropins are helpful in the chloroplast movement. This is one point. Phototropins involved in the stomatal movement. Phototropin involved in the hypocortical elongation. Phototropin involved in the phototropism. These four are the major function. One is chloroplast movement, stomatal movement, inhibition of hypocortical elongation, phototropism. Now we are going to discuss one by one point very elaboratively. Among all, this point is one of the most important point, friends. Every students pay attention and just listen to me carefully. All the students understand. We human beings, when we exposure to the high intensity of the sunlight, but friends, when we exposure to the high intensity of sunlight, we will try to escape, we will try to move away. But what about this particular plants? Do you think plant can move like animals? No, it cannot move. So they have to adjust, they have to tolerate that high intensity of light condition. Now every student listen. While studying the photosynthesis process, we all people studied a mechanism called as a photoprotective mechanism. I hope every student remember. Friends, what is the general mechanism? Imagine everyone, I am a chlorophyll molecule. What is my function? What is my function? I will absorb the light energy and I will absorb the light energy into chemical energy. By that plant prepare the food. But everyone understand how much amount of the light energy I can absorb? Only a limited amount. Only a limited amount of light energy I can convert it into chemical energy. That's all. But if the light is continuously coming from the sun, I can't convert entire light energy into chemical energy. I may die. So I am a chlorophyll, I may die because I can't convert the entire light energy into chemical energy. Now, every students listen to me carefully. Look into the concept. This is the dark condition. This is a weak blue light. This is the strong blue light. See friends, darkness. See, you are all able to see now, friends. These are all chloroplasts. These are all chloroplasts. They are not at all moving. See here, chloroplasts are not moving. But friends, whenever they expose it to the blue light, you know, see here, there is a movement of the chloroplasts are started towards one edge. Now all the students look into this beautiful picture. Once they expose it to the strong blue light, friends, they are all, you all people look into this. They all are, every one of you listen to me. They all are moving towards an edges. Let me explain you very elaboratively. Friends, all the students listen to me carefully. How does chloroplasts are arranged actually like this. Like this, chloroplasts are there. These are all the chloroplast molecule. Now everyone please listen my words carefully. Now there is a high intensity of sunlight, very high intensity of sunlight. As a result, what happened? Most of the chloroplasts get damaged. So to avoid the damage, to avoid the excess amount of the sunlight, you know what these chloroplasts are doing? Look into this. Instead of arranging in a group, they are arranging in a layer. So this is one chloroplast, second chloroplast, third chloroplast, fourth chloroplast, fifth chloroplast, sixth, like grana. They are arranging. As a result, what happened? This is the point you have to understand. Now whenever there is a high intensity of sunlight which is getting damaged, only upper chloroplast will get damaged. Otherwise, imagine the second chloroplast also getting damaged, but remaining all the chloroplast, they will not be affected by the high intensity of sunlight. In a simple way, what I would like to conclude here is that whenever plants exposed to the high intensity of sunlight, in order to protect the chloroplast, with the help of phototropins, chloroplast starts moving towards the edges and chloroplast are arranging in the form of a layers. When the chloroplast are arranged in the form of a layer, only upper chloroplast can get affected completely. The lower chloroplast never affected. So how does, see, this is the arrangement, right? Initial arrangement. How they are moving like this, how they are arranging like a layer, how is that possible? That is possible exclusively because of the phototropin. What the phototropins will do? They will stimulate the strong blue light. That strong blue light only, friends, will allow the chloroplast to arrange in the form of a layer. Kindly do not confuse. If they ask you a question in the examination saying that which of the following photoreceptors plays a key role in the chloroplast movement and the stomatal closure and chloroplast movement only mediated by the phototropin. Cryptochrome and phytochrome do not have any direct role or indirect role either in opening or closing of the stomata or in the movement of the chloroplast. Keep that points in your mind without fail. Okay, so that is the first point. What's the second point? Stomatal movement, inhibition of hypocortical elongation. The most important function where we are going to get the experimental based questions. I told you now, there is a great way that we can expect the questions experimental, that is only from here. Now stomatal movement. So friends, every student commonly know that. What is the major function of the stomata? Stomata is exclusively responsible for gaseous exchange. Stomata is exclusively responsible for transpiration or the water loss. Major is what? Gaseous exchange. Now we also know very well, friends, we read many times, when the stomata will be opened, when the stomata will be closed. We all people know, right? When the guard cell become turgid, stomata will be opened. When the guard cell become flaccid, stomata will be closed. Just a simple. Now you can ask me, how does turgidity occur? How does flaccidity occur? We all people know that we are reading from the 10th standard exclusively because of the potassium. Majorly potassium, water, chlorine. Every student listen. When abundant amount of the potassium, abundant amount of chlorine, abundant amount of water molecules, when they are all enter into the guard cell, what happens, friends? Guard cell will become swollen, which we can call turgid. It created turgor pressure. Because of that particular turgor pressure, what is happening, friends? The guard cell is getting open. Now you only tell me what's the opposite. Whenever, friends, listen my words carefully, whenever abundant amount of potassium, chlorine, water, they are not going inside, they are all coming out. They are all coming out of the Guard cell. Once all the ions come out of the Guard cell, what happens? Guard cell will become completely flaccid. Because of the flaccidity, what is happening, friends? The stomata is getting closed. Very simple. Turgidity and flaccidity. I hope every student remember while discussing the hormone abscisic acid, we all people studied on the Guard cell membrane there are receptors are there, yes or no? To that receptor who will come and attaches? Abscisic acid only come and attaches. Abscisic acid is the major major hormone which is involved in pumping water, chlorine, potassium out of the cell, and it is involved in the closure of this particular stomata. Kindly don't confuse with this. Okay. Now, what are the various factors affecting the rate of transpiration? These are all very unique statement, my friends, and easy statement. Water stress. Low water availability cause the stomata to close. There is no doubt about it, because already inside, listen to me carefully, already inside the plant body, very limited amount of the water is present. Very limited amount of water is present. Now that all water go out, plant may die. So when the water concentration is low in the plant body, what abscisic acid will do? It will get released. I hope you all people know very well. What do we call it? The abscisic acid. We call the abscisic acid as the stress hormone. So it is releasing in the stress condition. It will allow the plant to close the stomata. Once the stomata get closed, more amount of the water never lose. Day and night. I hope every student remember this. Where we studied this? In the camp plants. What is the speciality of a camp plants? They will open the stomata night time. They will close the stomata during daytime. I hope every student remember. Whatever the stomata that is opening in night time, what do you call it? That particular stomata? Sunken stomata. What do you call, friends? Sunken stomata, which opens only during the night time. What happens when they open during the night time? They will prepare the food, they will store the food. During daytime, they won't open any stomata, but they will use the food. During the daytime, they close the stomata. As a result, what happened? The water will be conserved completely. Because camp plants means what kind of plants? All the camp plants are desert plants. What is the temperature in the desert? In the desert, during the daytime, the temperature is very higher. But when it comes to the night time, temperature dramatically fall down. It's very cool. As a result, what happened? Daytime, they should not open the stomata. So that's why they open the stomata night time. They close the stomata during daytime with for the purpose of conserving the water. That's a simple point. Now, carbon dioxide levels. High carbon dioxide level cause the stomata to close. Friends, this is really a good point. You all people know what is the major function of the stomata. We discussed the major function of the stomata is gaseous exchange. We all people know very well. When the carbon dioxide enter inside, what happens? Plant use that carbon, convert into sugar. They will use for their growth and development. So already inside, listen my statements carefully. Inside the plant body, already abundant amount of the carbon is present. Then why once again we have to bring the new carbon inside? No, there is no requirement. But inside the plant, there is no carbon. Then obviously we have to open the stomata. So what's the statement? High carbon dioxide level cause the stomata to close, while low levels cause the stomata to get opened. Right. Friends, is that clear everyone till this slide? Please let me know. And we are having only one concept now, photoperiodism. I told you now, from here we will get the experimental based questions. This kind of questions. Very common questions and easy questions. We will see in the next class. Is it clear, everyone? Okay. So friends, listen to me. Everyone think and let me know. So what I am planning is, everyone think and let me know. You don't need to worry about it. So today evening, that is from, let's say, consider, friends, just listen to me carefully. If everyone is willing to attend, then only I will take the class. Otherwise I don't take it. Okay, just listen. Today evening, which unit we are going to start next? Sumaya, I am going to start unit 1. Okay, listen. Today evening, see, there is a little concept of this particular photoperiodism is left. It will just take 20-25 minutes. I'll finish it. After that, I want to solve all the papers of the CSIR, especially 21, 20, 19, 18. At least I want to solve all these papers, okay, in front of you. So what I am planning, I don't know whether you are habituated to this kind of thing or not. I will take today itself 4 hours of class, at least, if not 4 hours, 3 to 3.5 hours. So which is midnight. If you are all ready, then only I will take. So 10:30 to 1:30 or 2 p.m. sorry 2 a.m. I will take, because today is Saturday. So by that tomorrow you can take the rest. You can sleep happily. If everyone is ready, you just think and let me know in the Telegram channel. By that we can conduct the class for 4 hours of time. By that I'll finish the entire practice question, sorry, PYQ questions from the plant physiology. If you are fine, then only. Otherwise you no need to worry about that. We will see in the normal classes only. Okay, you people think and let me know in the Telegram. By that we'll schedule the class for 10:30 p.m. Okay, minimum 3 hours at least I will take. By that we can practice more number of questions from the previous year papers. Okay friends, that's all for now. I want you to decide and let me know. Based on the majority of strength I will take, majority of responses. Okay friends, that's all for now. I'll see you in the next class. Bye bye.